Good morning, everyone. Good morning, sir. How's your morning, guys? I hope you had a great sleep and also a wonderful morning. So, don't forget to take your breakfast because it's another day and another time to learn something about biochemistry. So, just for checklist, we already finished tackling about glycolysis, the Krebs cycle, oxidative phosphorylation, beta oxidation, gluconeogenesis, and glycogenesis, respectively. And now we are about to learn the pentose phosphate pathway. Okay, does it sound something new to you guys? Because it does to me the first time I heard of it. Okay, so but worry not because we will know further about this topic in the next few minutes. And this topic will be discussed by our group, which is group number 7. Okay, so before anything else, I would like you all to meet the team, which is composed of 4 lovely ladies, namely Aiden Marie Carion, Princess Anne Encarnacion, Joshin Limanog, and yours truly, Nina May Sanchez. Okay, so just for transparency, everyone, written here are the contributions of each member on today's presentation. So first, for Edin Marie Carion, she contributed on the presentation or the slides, the audio presentation, of course, and she is assigned to discuss about one of the 2 phases of the pentose phosphate pathway, the oxidative phase. Next, Princess Anne Encarnacion, she contributed also on the PowerPoint presentation, the audio presentation, and the reporter assigned for the non-oxidative phase. Next, Joshin Lumanog is assigned to report about the regulation of pentose phosphate pathway, and she also contributed on the presentation slides and also the audio presentation, of course. And yours truly, Nina May Sanchez, also the PowerPoint presentation slides, the audio presentation I am doing right now, and I am assigned to report and introduce to you all the pentose phosphate pathway. Okay, so now I am done introducing the members of the team. So now let's proceed to the table of contents. So these are the topics that you will learn later on, which will be discussed by the members mentioned earlier. So here written are the pentose phosphate pathway, the oxidative phase, non-oxidative phase, pentose phosphate pathway regulation, and lastly, its clinical relevance. Okay, so let's proceed to the next one. So let's proceed to our objectives this morning. So number 1, to be able to understand the function of the pentose phosphate pathway in the production of NADPH and ribose precursors for the synthesis of nucleic acid. Number 2, to identify and demonstrate the 2 phases under the pentose phosphate pathway. And number 3, to gain knowledge on how the regulation of the pentose phosphate pathway works and what this is for. Okay, so next, the question here is, what is pentose phosphate pathway? Or what you will learn in this topic later on. Okay, so now let's have its topic outline or the topic outline. So we will discuss introduction. Of course, we will introduce what this pathway is, so its location, function, and its 2 phases. So for the phases, it has oxidative and non-oxidative, and lastly, the regulation and clinical relevance. So role of NADPH in regulating the partitioning of glucose 6-phosphate between glycolysis and the pentose phosphate pathway. Alright, so now let's officially start our lesson. So DNA and RNA are 2 types of genetic material that are found in every cell of every living organism. So these genetic materials serve as blueprints. So these instructions on how to construct the protein structures that are the building blocks of all living things that are necessary for life, because they include the data needed to produce those protein structures. So when DNA and RNA are first manufactured, there are multiple layers of quality control to help avoid and repair mistakes. This is because the structural and functional significance of DNA and RNA for all living things demands multiple layers of quality control. And there is an additional pathway that can be taken during glycolysis that results in the production of sugars that are used in the synthesis of RNA and DNA. So this important physiological mechanism is known to us the pentose phosphate pathway, which is also sometimes referred to as the hexose monophosphate or HMP shunt. So this pathway, and also it's also called as phosphogluconate pathway. So this pathway is unique in that it does neither produce or consume any energy in the form of ATP or adenosine triphosphate. And this makes it one of the most important pathways in every organism. And in the process of metabolizing glucose, this is also an alternate pathway. It has a more anabolic quality to it, and it is important to note that this pathway or this process occurs in the cytosol of the cell. So NADP plus serves as the electron acceptor in this certain oxidative pathway, which ultimately results in NADPH being produced. Pentose ribose 5-phosphate is utilized by rapidly proliferating cells in order to produce RNA, DNA, and various coenzymes such as ATP, NADH, FADH2, and coenzyme A. So these cells can be found in bone marrow, skin, and the intestinal mucosa. So also during HMP shunt, highly active tissues include the liver, adipose tissue, adrenal gland, erythrocytes, testes, and lactating mammary glands. So we should also note that this pathway can take place in one of two phases, the oxidative phase and the non-oxidative phase. So during the oxidative phase, glucose 6-phosphate is utilized in order to make nicotinamide adenine dinucleotide phosphate or the NADPH, and ribulose 5-phosphate, which can be converted to ribose 5-phosphate. And the next one is the non-oxidative phase, which consists of a collection of various reversible processes. So the intermediates produced during these reactions are connected to a number of different pathways, such as glycolysis, the synthesis of aromatic amino acids, and the synthesis of nucleotides. So this pathway, the pentose phosphate pathway, leads to the production of intermediates that can serve a variety of functions. Okay, so now let's proceed to the next slide. So its biological significance of the intermediates. So first is the NADPH. So it is used in the synthesis of certain amino acids, and also it is used for biosynthesis of fatty acids and steroids. Next, the ribose 5-phosphate. So it is an essential component in the process of nucleotide biosynthesis. Next, the third one, erythrose 4-phosphate. It is used for the processing of aromatic amino acids, and it also includes other metabolites such as for glycolysis and gluconeogenesis. Okay, so now I am done introducing the pentose phosphate pathway. So now let's have its two phases, which is namely the oxidative phase and the non-oxidative phase, which will be further discussed by Edin Marie Carrion and Princess Anne Encarnacion. Okay, so you can now take the virtual stage.
Thank you, Miss Nina. Good day, everyone. I am Edin Marie, and now we are going to learn the oxidative phase of the pentose phosphate pathway. The oxidative in this phase comes from the oxidation process, which, as we recall, is the loss of electron as an interaction between contacts of two or more elements. The oxidative phase makes up three major steps wherein two oxidation processes are irreversible and there is also the hydrolysis. We will be able to identify these processes as we learn the reactions consisting this phase. Now, this phase mainly takes place in the cytosol, which we know is the liquid found inside cells. To acquaint you, the oxidative phase serves as the first stage of pentose phosphate pathway, starting with the glucose 6-phosphate converted into ribulose 5-phosphate that then produces NADPH and CO2 or carbon dioxide molecule from the released carbon 1 of the glucose 6-phosphate. To understand better, let us have a quick review on the initial molecules and products of the oxidative phase. The initial molecules include the glucose 6-phosphate, which is the first intermediate of glucose metabolism, having a pivotal role as a substrate of the liver during energy metabolism. In the structure, we can notice that this glucose sugar is phosphorylated at the hydroxyl group on carbon 6. Next is the NADP+, that comes from anabolic reactions. NADP+ is a coenzyme that functions as a universal electron carrier, accepting electrons and hydrogen atoms to form NADPH. And lastly, the water molecule or H2O that contains two hydrogen atoms joined by a single oxygen atom. Next are the significant products at the end of the phase. The ribulose 5-phosphate, which is both a product and intermediate, easily converted to ribose 5-phosphate to synthesize nucleotides and nucleic acids. Then the reduced nicotinamide adenine dinucleotide phosphate or NADPH, which is an essential electron donor in all organisms and provides the reducing power for anabolic reactions and redox balance. Lastly, the carbon dioxide, which was from the cleaving of carbon 1 in glucose 6-phosphate. Now, let us proceed to the reactions within the three major processes of the oxidative phase. Let us also take note on the involvement of enzymes within the reaction mechanisms. So let us have a look on the image on the side of the text. In the first step of the oxidative phase, glucose 6-phosphate dehydrogenase catalyzes the oxidation of glucose 6-phosphate to 6-phosphoglucono-delta-lactone, an intramolecular ester, via the transfer of a hydride ion from carbon 1 of glucose 6-phosphate to NADP+, that acts as an oxidizing agent. So what happens is, we have this NADP+ molecule that will act as an electron acceptor, so we essentially extract two electrons from this particular glucose 6-phosphate, and those two electrons are picked up by this molecule. In addition, hydrogen ion is picked up by this molecule as well, and we generate the reduced version of nicotinamide adenine dinucleotide phosphate. So, dehydrogenation simply means we're removing hydrogen ions, so an enzyme called glucose 6-phosphate dehydrogenase catalyzes the transfer of a hydride ion. So that's a hydrogen ion and two electrons from the glucose and onto the end of the NADP+ carrier molecule. So this is the electron acceptor, as we remember, that picks up two electrons and that hydrogen ion. Then we also release a hydrogen ion, so now we have the 6-phosphoglucono-delta-lactone. This reaction yields the first molecule of NADPH of the pentose phosphate pathway. This step is an irreversible step of oxidation and a rate-limiting step, so this means that this step has the greatest activation energy and therefore the step that has the greatest influence on reaction rate. Following the oxidation of glucose 6-phosphate, another reaction catalyzed by a different enzyme uses water to form 6-phosphogluconate, which we will know now. So this is in the second step of the oxidative phase. The 6-phosphogluconate delta lactone is hydrolyzed to 6-phosphogluconate, a linear molecule. 6-phosphogluconate delta lactone is hydrolytically unstable and undergoes a non-enzymatic ring opening, a reaction that occurs at a significant rate. However, in the cell, this ring opening reaction, hydrolysis, is accelerated by the catalytic action of 6-phosphogluconolactonase, which is the enzyme that helps this step. In the third step of the oxidative phase, now the 6-phosphogluconate undergoes an oxidative decarboxylation to form the ribulose 5-phosphate. This is a ketopentose, and to also form carbon dioxide and a molecule of NADPH. To oxidize the 6-phosphogluconate, another molecule of NADP plus picks up electrons. So, carbon 1 will be removed, forming the carbon dioxide, and 2 electrons along with a hydrogen ion that is extracted from the sugar molecule and placed to the NADP molecule, therefore forms the ribulose 5-phosphate and an NADPH molecule. The reaction is catalyzed by 6-phosphogluconate dehydrogenase enzyme that requires the presence of magnesium ions. This reaction notably yields the second molecule of NADPH of the pentose phosphate pathway. Therefore, the oxidative phase of the pentose phosphate pathway ends with the production of ribulose 5-phosphate, namely the substrate for the reactions of the non-oxidative phase. And so, as we recall, we can now identify that the two irreversible reaction mechanisms are actually the first and the third major steps of the oxidative phase, where we have hydrolysis in the second step. Finally, as an overview, in step 1, we have undergone an oxidative reaction and oxidation reduction reaction, which we generated that first NADPH molecule. In the second step, we opened up this molecule to basically be prepared for step 3. Because in step 3, we want to generate yet another NADPH molecule as well as release that carbon dioxide to generate a pentose sugar. And this ribulose 5-phosphate will then be transformed into its isomer form, the ribose 5-phosphate, which can then be used in the second phase of pentose phosphate pathway, the non-oxidative phase that we're going to focus on in the next presenter.
So thank you, Adan. And now my name is Princess Ann Encarnacion, and I will be discussing the non-oxidative phase of the pentose phosphate pathway. So as previously mentioned, the first phase of the pentose phosphate pathway generates 2 NADPH molecules and a pentose sugar called ribose 5-phosphate. However, when the cell needs NADPH more than ribose 5-phosphate, the cell can convert the ribose sugar into specific glycolytic intermediates via the non-oxidative phase, which is the second phase of the pentose phosphate pathway. So, the non-oxidative phase is a collection of several reversible reactions in which the intermediates are connected to several other pathways, including nucleotide synthesis, aromatic amino acid synthesis, and glycolysis. The ribulose 5-phosphate can undergo a series of isomerizations as well as transaldylations and transketylations that result in the production of other pentose phosphates. So the overall net reaction of this phase transforms 3 ribose 5-phosphate molecules into 2 fructose 6-phosphate molecules and 1 glyceraldehyde 3-phosphate. So the non-oxidative phase can be broken down into 4 processes. So let's begin by focusing on process 1. Now in process 1, we actually have 2 steps. In the first step, we want to take the ribose 5-phosphate molecule and transform it into an isomer version, which is the ribulose 5-phosphate. So the phosphopentose isomerase here, as you can see, catalyzes this reaction. Once we form the ribulose 5-phosphate, it then undergoes a second reaction, which is catalyzed by the enzyme phosphopentose epimerase. So what this does is it transforms the ribulose 5-phosphate into cellulose 5-phosphate. And the only difference between these two molecules is the stereochemistry of the third carbon, which is highlighted in blue color. Now, why do we need the specific stereochemistry in cellulose? Why can't we use the stereochemistry in ribulose? This is because the enzyme in the second process of this pathway basically uses only the type of stereochemistry in cellulose. So ultimately, in process 1, we want to transform the ribose 5-phosphate into cellulose 5-phosphate. And notice that we begin with two of them. So we have two of the ribulose intermediates, and then we form two of the cellulose 5-phosphate product. One of these cellulose molecules is used in process 2, and the other one is used in process 4, which will be discussed later. Let's move on to process number 2. So we take one of the cellulose 5-phosphate molecules, and then we take another ribose 5-phosphate molecules. So far, we actually used 3 ribose 5-phosphate. 2 were used in the process number 1, and now the third one is what we are using in this particular step. So we have the enzyme transcatolase, which actually requires a cofactor we call thiamine pyrophosphate. Basically, what the transcatolase actually does is it takes a 2-carbon group from the cellulose. So here we will take the entire section, which is the, as you can see, there is the OCCH2OH at the top of the cellulose 5-phosphate, and then it will be placed into the ribose, the OCH at the top of its stereochemistry. So notice both of those are pentose sugars, 5 carbon sugars, and then when we transfer the OCCH2OH of the cellulose 5-phosphate into the COH part of the ribose 5-phosphate, we basically extend the sugar by 2 carbons, and we form a 7-carbon sugar known as pseudoheptulose 7-phosphate. Now, if we take away the 2 carbons from the cellulose, from the CCH2OH part, we essentially form a 3-carbon sugar, and that 3-carbon sugar is known as glyceraldehyde 3-phosphate. So the products of process number 2 reaction of this particular phase are the pseudoheptulose 7-phosphate and the glyceraldehyde 3-phosphate, and is catalyzed by the enzyme transcatolase. In process 3 of the non-oxidative phase, we now take the pseudoheptulose 7-phosphate and the glyceraldehyde 3-phosphate, and these two products will act as reactants in the process number 3. Now, the enzyme in this particular case will be different. The enzyme in process 3 is transaldolase, and what transaldolase does is it takes the entire carbon section from pseudoheptulose 7-phosphate. So that would be the, as you can see, the HOCH part until the OCCH2OH part at the top. And then it transfers it into the carbon from glyceraldehyde, the OCH at the top. So we transform pseudoheptulose molecule into erythrose 4-phosphate. So we take off 3 carbons from pseudoheptulose, and so erythrose is a 4-carbon sugar, and then we place the 3 carbons into the glyceraldehyde molecule. So we form a 6-carbon sugar called the fructose 6 phosphate. And this fructose 6 phosphate is one of the glycolytic intermediate products. So the fructose 6 phosphate is actually the final product or one of the final products in the non-oxidative phase. What we want to do in the final step in the final process is we want to take the erythrose 4 phosphate produced in process number 3, and we want to take the second cellulose 5 phosphate that we still have left over from process 1. So remember that we only use one of the 2 cellulose molecules that we produced in process 1. So we use one in process number 2, and now we use the second one in this process number 4. Now, the enzyme that catalyzes the process 4 is the same enzyme that we used in process number 2. So it's no surprise that this same enzyme uses the same substrate molecule, which is the cellulose. And what it does is, just like in the process number 2, it took off the 2 carbon component from the cellulose and placed it into the ribose. So here, once again, we take the 2 carbon from cellulose, so and that would be the CH2OH part at the top, and then we place it into the sugar molecule in erythrose. And so we extend the 4 carbon sugar in erythrose 4 phosphate by 2, and we form a 6 carbon sugar that we call fructose 6 phosphate, which is once again a glycolytic intermediate molecule. And then when we cut the CH2OH part of the cellulose 5 phosphate by 2 carbons, we form the 3 carbon triose molecule known as GAP, or the glyceraldehyde 3 phosphate. And remember that GAP, or glyceraldehyde 3 phosphate, is also a glycolytic intermediate. So we see that if we sum up all the 4 processes, the net reaction that we actually get is we have the input 3 ribose 5 phosphate molecules, so 2 in process 1 and 1 in process 2. We essentially get back to fructose 6 phosphate molecules and a single glyceraldehyde 3 phosphate. So now these are the glycolytic intermediates that can be transformed into glucose 6 phosphate. And that glucose 6 phosphate can undergo the oxidative phase to form even more NADPH molecule. So basically, when the cell needs the NADPH much more than it actually needs the ribose 5 phosphate, we undergo the non-oxidative phase of the pentose phosphate pathway to help us generate even more of these NADPH molecules. On top of that, what the non-oxidative phase of the pentose phosphate pathway allows us to do is it allows us to actually ingest ribose sugars into our body, because via this particular pathway we can actually break down the ribose sugar into glycolytic instruments, and then those intermediates can be used via glycolysis to actually generate high energy ATP molecules. So here we see that the link between glycolysis and the pentose phosphate pathway is the non-oxidative phase of the pentose phosphate pathway. So that would be all for this topic. Let's move on to the pentose phosphate pathway regulation.
Very well explained, Miss Encarnacion. So again, everyone, what are the 2 phases of pentose phosphate pathway? Okay, yes, it's oxidative and non-oxidative phase. Hello everyone, this is Joshin, and now let us move on to the next discussion, which is the regulation of pentose phosphate pathway. So basically, at the end of our discussion, we'll probably identify how pentose phosphate pathway is regulated. But prior to that, let us first have a short recall of the previous topics. So while the products of glycolysis are sent through the remainder of cellular respiration to produce energy, or before we go through the citric acid cycle, an arrow pointing to the pentose phosphate pathway can be seen in the diagram. Am I right? Okay, so what is this pathway? Is this still a part of the cellular respiration process, or does this route result in the generation of more ATP? So basically, this route or pathway is an alternative branch from glycolysis that generates the sugar that comprise DNA and RNA. This is a unique mechanism since neither ATP nor adenosine triphosphate is created nor consumed. So once glucose is in the cell, an enzyme called hexokinase attaches a phosphate group to its sixth carbon, creating glucose 6-phosphate. So from there, the cell has the option to take glucose through a metabolic pathway called glycolysis, which is the breakdown of glucose to generate ATP. However, if the cell doesn't need ATP, glucose can be used to make more other useful products by just entering in an alternative metabolic pathway called pentose phosphate pathway. So, glucose can enter this pathway when cells need to make ribose or NADPH. Again, in this pathway, it oxidizes glucose to form NADPH and other carbohydrates for biosynthesis, and it does not generate or use ATP like glycolysis does. And ribose can be used to make nucleotides, which are the building blocks of our DNA and RNA. And just like glycolysis, the pentose phosphate pathway happens exclusively in the cytoplasm, and it doesn't require any special organelles, which means that all of our cells can use this pathway. Okay, so when the demand of NADPH slows, the level of NADP plus drops. So if the ratio is low, that means reducing equivalents are plenty or high in the cell. Then there is no need for glucose 6-phosphate dehydrogenase enzyme to be active, or there is no need for pentose pathway to be active, while the glucose 6-phosphate molecule is instead used to fuel glycolysis. On the other hand, when the NADPH level is high, when the NADPH ratio is very high, it means that the NADP plus concentration is high, which means the reducing equivalence is low, and when it is low, we can promote pentose phosphate pathway. So this is the point of determining factor in regulating pentose phosphate pathway. So to elaborate, low levels of NADP plus inhibit the dehydrogenation of glucose 6-phosphate because NADP plus required as the electron acceptor, and the NADPH competes with NADP plus in binding the active site of the enzyme. Okay, to sum it up, it is stimulated by NADP plus and glucose 6-phosphate while inhibited by NADPH, or the key negative feedback mechanism, and the acetyl coenzyme A. While the entry of glucose 6-phosphate either into glycolysis or into pentose phosphate pathway is largely determined by the relative concentrations of NADP plus and NADPH. Okay, lastly, we have here the clinical relevance. So we'll be further discussing G6PD deficiency, malaria, and tranaldolase deficiency in the next pages, and on how they become related to our main topic for today's lesson, which is the pentose phosphate pathway. Okay, the first one is the G6PD, or the glucose 6-phosphate dehydrogenase. So it is a type of intravascular hemolytic anemia inherited in an X-linked recessive manner. So according to sources, it is the most common genetic enzyme disorder in humans. A deficiency in G6PD results in an insufficient amount of NADPH, and without which glutathione cannot be reduced in RBCs. So these red blood cells are not protected from oxidative stressors, resulting in damage and hemolysis. Okay, so next is malaria. I think we are all familiar with this disease. It is an infectious parasite disease caused by the Plasmodium genus, which is serious and can be fatal. Usually, malaria patients generally have high fevers, shivering chills, and flu-like symptoms. So a deficiency in G6PD can potentially protect individuals from malaria since these parasites require NADPH, and NADPH is probably produced by G6PD enzyme. Okay, wow, we're almost done with our topic. So let's proceed to the last one, which is the tranaldolase deficiency. So it is an inborn error of the pentose phosphate pathway that is present in the neonatal or antenatal period that affects metabolism with hydrospitalis, hepatosplenomegaly, liver dysfunction, congenital heart disease, and dysmorphic facial features. So we have here an illustration that shows an example of hepatosplenomegaly, a disorder where both the liver and spleen swell beyond their normal size. So if we try to compare it physically outside our stomach, we can actually see the difference compared to the given illustration. Okay, so I believe that will be all for today's discussion. Again, I am Joshin, and we hope that you guys learned something from our discussion. Thank you, and God bless you all.